Hey guys, we are going to go ahead and do the last lecture in this module, which is on infectious disease. Okay. All right. So this first one, disease-causing agents. These are pathogens. Then we have, the next one is bacteria these are prokaryotes. Prokaryotes are a group of single-celled organisms. They don't have a nucleus. Camera's weird. They don't have a nucleus and they lack membrane-bound organelles, really with the exception of ribosomes. Bacteria commonly come in three shapes. We have the coccus. Rod-shaped is bacillus. Oh shoot, I don't remember how many L's there are in this. I think it's one. That doesn't look right. No, it's because there's two. It's one of those words I always have to look at. And spiral shaped, which are just called spiral. Okay. All right, so we need to label these or fill in the structures. So this first one is going to be our cell wall. I'm going to protect the bacteria. Contains something called peptidoglycan, and it allows for classification. And those classes are gram-positive and gram-negative, respectively. And they are classified based on basically the thickness of the cell wall. Then we have the capsule, gelatinous layer outside of the cell wall of some but not all bacteria. It doesn't have any surface proteins, which means that the immune system has a harder time identifying it, kind of slides under the radar. Okay. This should be flagella. It's that whip-like projection right here. Rotates 360 degrees and allows the bacteria to move in its little environment. Then we have these stiff fibers are called fimbria. Allows some types of bacteria to stick to other cells, okay, like host cells. We have the pilus. Elongated hollow appendage used to transfer DNA from one cell to another. So it's shown here. A pilus is essentially a reproductive structure, if you can really call it reproduction. Bacteria don't do sexual reproduction, but they are able to transfer genetic information between cells, between individual bacteria, which is important because when we talk about bacteria becoming resistant to antibiotics, this kind of immunity develops from bacteria sharing their DNA, which would include resistance to some of these antibiotics. And they share that by exchanging that information through the pilus. And then, speaking of DNA, we have the plasmid. Circular DNA found in many bacteria. We talked about antibiotic resistance. The plasmid is separate, kind of, from the regular bacterial DNA. It's extra. And a lot of the adaptation and evolution, if you will, of a bacteria species comes from the plasmid. Okay? All right, let's move on. Bacterial classification. All right, we talked about the cell wall and having either gram-negative or gram-positive bacteria. So let's go ahead and look at that. Gram-positive bacteria have a thick cell wall. Let me move this out of the way. Okay, thick cell wall. Because of that, they tend to stain a really dark purple. And it's because the material that the cell walls are made out of absorb the stain that's used really well. Okay, so they turn really dark. Gram-negative bacteria have a thin cell wall. So they don't stain as dark because there's not as much of that to react with the stain itself. So you tend to get a light pink. And the images here that have these samples of gram-negative and gram-positive bacteria, that's accurate. This is what they look like under a microscope. These appear to be cocci, or the round ones. And these appear to be bacilli, the rod shape. And adding the IFE just makes it plural. Okay, so gram-positive and negative bacteria have different treatments. This is really true because some bacterial treatments go after the cell wall. For instance, the psyllin drugs, penicillin, amoxicillin, ampicillin. All of those are actually like enzyme inhibitors that interfere with the ability of bacteria to build cell walls. Without that ability, they can't build, or they can't reproduce, if you will, right? The bacteria can't produce more bacteria. So these antibiotics are going to inhibit bacterial growth. However, for gram-negative bacteria, you've got to go with a different class of drugs, ones that target other parts of the bacteria rather than the cell wall, because in gram-negative bacteria, the cell wall is just not thick enough and doesn't play the same type of role that it does in gram-positive. Okay, why do we refrigerate things? It helps to slow the growth of bacteria. It's not going to stop it. That's why your food can still get nasty in the fridge if you've left it in there for too long. But it does slow it down. Okay, viruses, generally considered to be non-living. Generally because there's, a group of scientists that feel that viruses should be considered living. However, classically, they don't fit the requirements associated with living things. They have to be inside of a host to replicate. They don't have their own machinery to let them replicate. That's one of the reasons. They're not composed of cells. Okay. In addition to not being able to replicate without a host, they also can't have any nutrients without a host. So they're very, very host dependent. And those two things, not having a cell and the host dependency, have continued to prevent them from being classified as living. Okay. The reason why people, some scientists would like to have it classified as living is because the virus's ability to adapt is quite impressive. It's why you have new strains of viruses like COVID-19. It's a coronavirus, that's the family of viruses it belongs to, but viruses can mutate depending on their environment, the host, and available resources. When they mutate, you get a different serotype. Okay, so you have all kinds of different coronaviruses, right? SARS was one, MERS was one those are back a minute. And then coronavirus, and just it's kind of what do I want to say? When it's not in its COVID, MERS, or SARS form, it has symptoms that are like the common cold. So anyway, they can mutate and evolve, and it's so impressive. Their abilities are so impressive to do that. They've been around since probably the beginning of humanity, and yet we've yet to eradicate them. We don't even have a cure for the common cold because it keeps changing. Your flu shots you get every year generally are made of a different mix of viruses because different seasons bring different forms of the virus. So that's why some want it to be considered. It's one of the reasons why some people want it to be considered living. All right, so viruses have something called a capsid, and it's just a protein container for its genetic material. Tupperware for DNA. Okay. The capsid has specialized spike proteins that help it attach to the host, to the host cell. So actually, I'll get there in just a second, hang on. In viruses, genetic material can be either DNA or it can be RNA, which means it can also be single-stranded or double-stranded. And viruses are classified partly based off of if they're DNA, if they're RNA, if they're single or double-stranded, their mode of reproduction within a host cell. Okay, so let's go ahead and look at these images real quick. And I'm going to go over a couple of other terms that we don't really have outlined here, but this is an adenovirus, and this is an influenza. Influenza, a lot of times when we talk about the word flu, we tend to think about like the stomach flu, throwing up and all the awfulness that way. Influenza, like influenza A and B, the type of influenza is the swine flu that we get vaccinated for in flu shots. Those are respiratory viruses. They can have like intestinal components as far as symptoms are concerned, but when you get a flu shot, you're not being vaccinated against like the stomach bugs that go around, again, with all the throwing up and stuff. Okay, so both of these types of viruses can cause similar symptoms, but notice the differences in their structures. So the adenovirus has our kind of, it's very geometric shape when it comes to the capsid, and it does look like this. I mean, this is a colorized drawing, but the shape looks like this under a specialized microscope. It's pretty cool. And then of course we have our spike proteins, right, that are going to help it attach to the cell, the host cell, so that it can start its invasion, kind of like a Trojan horse. The influenza virus is different because in addition to having its capsid, which are these green dots in here, it also has something called an envelope. Okay, not all viruses have envelopes. The ones that don't are called naked capsid viruses. You don't technically need to know this for an exam, but I'm telling you because I really like virology. Naked capsid virus, okay, and then we have our enveloped viruses, which is what this is. So you've got the capsid still, but you've got another layer kind of membrane around it. Okay, so what happens, are antibiotics effective against viruses? The short answer is no, because they're not alive. Antibiotics, notice the word bio in there, referring to something that's alive. Antibiotics classify or target living things such as bacteria, fungi, parasites, stuff like that. But they're not going to work against viruses because they're not alive. That's why if you go in and you have a viral infection of something, They're not going to start you on antibiotics. There's no point, okay? Viruses that have symptoms that hang around longer can create a good breeding ground in the body for a secondary bacterial infection, which usually after 10 days of having virus symptoms, they can treat you for a secondary bacterial infection. So things like COVID and influenza A and B, and a lot of other viruses, you can get sick with a virus. But then the virus, especially the ones that like to produce a lot of mucus and grossness, that creates a really great environment in the lungs for bacteria to grow. And so you can end up with a primary viral infection that started the whole thing, and then a secondary bacterial infection like pneumonia that you end up having to be treated with antibiotic for. Same thing happens with sinus infections. If you've had a cold, you start to feel better, and then all of a sudden you're like sick all over again. It's usually a bacterial infection that's settled into the sinuses. KKKK. So no, they don't. They don't work with viruses. All right, let's classify these terms, shall we? We will start with an epidemic. So an epidemic is when a disease has more cases than is expected for a period of time. So you have more cases of the flu in North America during a flu season than you normally anticipate, than you normally expect to have. That's an epidemic. Actually, take this one. Okay. We go from an epidemic then to an outbreak. And an outbreak is an epidemic that's confined to a local region. Okay. So you can have an epidemic that's just affecting North America. That would be an outbreak. So in order to have an outbreak, you first have to have an epidemic, right? You have to have more cases than expected in a given amount of time. And then for it to become an outbreak, it's not just like, in St. George, it has to be across a global scale, or not global, forgive me, a wider scale. That could just be isolated to the state of Utah, or it could be North America. Okay. A pandemic is when an epidemic goes global, if you will. Okay. So this is a global epidemic, which is what we're dealing with COVID. And frankly, have dealt with throughout history. The Spanish flu was a pandemic, about 100 years ago, actually, and it was nasty, nasty, nasty. Polio was a pandemic in the 1950s. The list is huge. I know it feels like COVID is the only one in a long time, but it's not. They're common throughout human history. Okay, so life cycle of a virus. How do these viruses reproduce? I love that we call it a life cycle, even though they're not technically alive. Anyway, so spike proteins attach to surface proteins on host cells. That's the first thing that's going to happen. And these spike proteins fit, if you will, on host cell receptors like a lock and key. This is one of the other reasons why, again, some people would like to have viruses be classified as living, because they're really specific. Viruses have the ability to infect a specific type of cell in the body. And they do that because their chemical shape of their spike proteins match the chemical structure of receptor proteins on host cells. They're really great at surviving. When this happens, which by the way is shown right here in the attachment phase, when attachment takes place, we move on to fusion. And this is where the membrane of the virus fuses with the membrane of the host cell. This is the Trojan horse part, because now the host cell has inadvertently let in this virus. It has the right key, right? It has the key of stuff that the cell would normally be letting in, except this one is an intruder. And so once it's fused, it releases its viral genetic material, either DNA or RNA. If this is an RNA virus, the RNA is first made into DNA. So it's a backwards process. Normally, we have DNA that's transcribed into RNA and then into a protein. Well, in this case, we go in with RNA, meaning we have to backtrack. It's called reverse transcription, make DNA, and then we'll go ahead with the rest of the process, okay? When viruses are going to do this, they combine with host DNA in the cell. Okay? And then the viral proteins and the genetic material are made by the host machinery. All right. That's kind of why I call viruses hijackers, because it's what they do. They hijack the cell. And so because viruses don't have any organelles, they have no way to make proteins. So they get into the cell, they release their genetic material, they combine it with the host genetic material, and then they use the host ribosomes and ER and Golgi and everything else in there to make more virus. Okay, so it essentially shuts down the cell's replication of normal cellular products that are being made, because it takes it over to now make viral proteins instead. Okay, then we have over here is budding. Essentially, once the new viruses have been created, they get shipped back to the membrane, where they are released into the bloodstream and into the surrounding tissues, where they go and infect other cells and continue the process until the immune system can get up and stop it. Okay. So many antiviral medications target the virus's ability to do three specific things that are required for the virus to make more of itself. The first one is just to keep it from attaching in the first place. The second is, that looked like an H, that didn't help. Anyway, the second is to perform reverse transcription. Okay, And then the last way is it prevents it from integrating with the host cell's DNA. So if antiviral meds can prevent a virus from being able to do these things, that's just kind of it. The virus simply can't replicate. All right, how are viruses used to cure disease? Well, essentially, viruses are used as something called vectors. And I don't think we've had that term, but a vector is, I can't really say it's an organism. A vector, say it's a method of delivery. For instance, mosquitoes are vectors for things like malaria and West Nile. They carry the parasites. So they're the delivery method for those parasites that cause those two diseases, okay? So in this case, we're talking about a virus being the delivery method. But rather than having viral DNA that's going to go in and cause all kinds of problems, a type of gene therapy happens, or there's been genetic manipulation. And so you get a therapeutic gene, something that's going to provide a cure, okay? That therapeutic gene is going to be put into a virus, and then the virus is going to do what viruses do. It's going to attach to the host cell using the spike proteins. It's going to fuse with the membrane, going to release the genetic information that it's carrying, which in this case is good DNA. That DNA is going to incorporate itself into the host cell's DNA, and all of a sudden, we have the host cell producing the proteins associated with the therapeutic gene. So I think that they're using an example of damaged skin. So you can take skin cells from an affected individual and you insert the gene for correct skin cell production into a virus. And then the virus is then, in this case, it's done in vitro, introduced into the boy's skin cells that they took, and then the skin is used as a grafting procedure. Let's see if it produces what, or I'm sorry, let's see what example this one does. So this is essentially a sickle cell disease treatment by removing the stem cells that cause sickle cell, which I don't know if you're, did we do this? I did, it's been a minute. But we talked about stem cell earlier in the semester, or sickle cell disease early in the semester, being a result of a single mutation in the DNA code for hemoglobin that will carry oxygen. And so they can, down here, B globin rescue, or actually this is beta globin rescue. Scientists engineer a virus to deliver a functional copy of the gene that produces beta globin. The gene has been modified to present the sickle cell. Okay, so it basically has the correction for that single mutation in DNA. The virus carries the correct gene with the correct sequence of DNA. Pretty nifty, ain't it? Is this the last page? No, for some reason I thought that was the last page. Let's talk about HIV. So HIV stands for human immunodeficiency virus. and it infects helper T's. Remember helper T's? I called them the sidekick T. They're the guys that are going to be secreting some of the chemical signals needed for an immune response. So it eventually leads to a decline in the white blood cell population, and HIV then becomes AIDS, which is acquired immunodeficiency syndrome. Okay, HIV first. In the acute phase of HIV, the progression, the virus replicates really rapidly. So the steps we just watched about attaching to the cell membrane and then releasing its genetic information and, you know, mixing with the host cell's DNA, that happens very quickly in the acute phase, and yet the person is asymptomatic. I'm not going to spit it. Oh, okay. They're asymptomatic because at that point, the body is still producing enough T cells to keep up with the virus. Okay, that's why you're not noticing it. But then we hit the chronic phase, and the chronic phase is when symptoms start, because now our T cell count has dropped, our white blood cell count overall has dropped. Some of the common early symptoms associated with HIV include shingles, especially for those that have had the chickenpox virus or exposure to chickenpox, because again, that virus lives in the host and just kind of goes dormant or latent until conditions are perfect for the virus to emerge again, and then it just comes out as a slightly different set of symptoms. Other things are kind of basically flu-like symptoms. This is your cough, fever, body aches, diarrhea, stuff like that for no reason. Then we reach our full-blown AIDS stage. And this is where we have a drastic reduction in T cells. Okay, it plummets like severely. So on this graph, we can see HIV is being represented by this purplish plum-colored line, and the white blood cells, specifically the T lymphocytes, are the blue. So you can see how we have where in the acute phase, we still have a high number of our T cells, so it's keeping the symptoms of the virus from showing up. And then those T cells begin to decline. And as they decline, we hit the chronic phase where, again, we get the flu-like symptoms, shingles, things like that. And then finally, as the T cell count drops hugely, we end up with AIDS. Okay? I don't know why I made that a we thing. It's not a we thing, I guess. It's just, this is what the virus would do. Okay, and at this point, because the T cell count is so low, and remember the T cells are the assassins, and then we've got those three groups of T cells. We've got the helper Ts, sidekick Ts, right? Then we have our cytotoxic or killer T cells, and then the memory T cells. They're essential for that remembered immunity, and when we take those out, we lose our best weapons to fight infection. And so you become susceptible to all kinds of stuff like pneumonia and tuberculosis. And tuberculosis can be a big one. And it's something that has really been, tuberculosis is the number of people that have it on its own without something else associated with it, another disease associated with it, especially in more first world countries, has plummeted. It's dropped so much. It used to be something that was around. People died of it frequently. It was called consumption back in the day. So if you ever read, Charles Dickens, there might be some in Jane Austen that talk about consumption. I know Anne of Green Gables talked about consumption. The Wild West. Yeah, I'm thinking of all of the times in pop culture that they talked about consumption, and the movie Tombstone. It was tuberculosis, but that's what they called it then. Okay. And because, hey, who doesn't want to talk more about COVID-19 in this COVID-19 world we live in? Which, as I have expressed once or twice before, I'm pretty sick of now. I'm kind of just done with it and would like it to go away forever. But let's talk about how it works. COVID-19, again, type of coronavirus, begins as an upper respiratory infection and has symptoms that you would associate with the common cold. Okay, the pharynx, the larynx, the trachea, the nasal passages. In other words, sore throat, laryngitis, and runny nose. Okay, stuff like that, and in its various forms. And then the virus is going to invade the cells that make all of these things up, the cells that make up the upper respiratory system. Okay. And then most people have the ability to contain this virus. Or in other words, the immune system has the ability to fight it effectively, okay? Immune systems can contain it. Contain, not like it's in them. Contain as in keep it under control. Okay. I'm sure you've seen this in the news and everywhere else, but this is what a coronavirus looks like. It has been colorized so you can see the different surface proteins and spike proteins, but it's called a coronavirus. Corona means crown in Spanish, I think in Latin, the form of it in Latin. And because it has this halo of these proteins all the way around it, it looks like it's wearing a crown, okay? So an upper respiratory infection can then progress into a lower respiratory infection. And this is where you're going to have something called goblet cells targeted. And these goblet cells are produce mucus. And so you get an increase in mucus production. Isn't that an awful word? Mucus. My friend and I had, don't ask me why, I don't know why, but we had a debate while playing Phasmophobia the other night about what word was more disgusting, mucus or sputum. I went with mucus. Nasty, nasty word. My friend said sputum. I disagree. what would you vote for? This is the real big question of 2020. This is the real big issue. Do you vote for mucus or sputum as being the most disgusting word? I need to know. Okay, so increases mucus production and inflammation. And we learned that a severe inflammatory response by the immune system is not a good thing. Okay? All right, so the body can kick into severe immune response, which I just said isn't always a good thing because then we end up with even more inflammation. And the little alveoli, which are the, if you remember, the little tiny sacs at the site of the little tiny sacs, which are the site of gas exchange in the lungs at the end of the bronchioles, they fill up with fluid. We get pneumonia. And in this severe stage, this is where the ventilator is required to oxygenate the body because the lungs aren't doing it. They can't because they're of the response to the virus. Okay. I talked about SARS earlier because I said it was a type of coronavirus. Here it is, has a similar structure. So it talks about this a little bit more. You can read through some of the information, again, which we probably all are pretty aware of how it works at this point. But because you guys are pre-med or going into some sort of medicine or dental, you have a better understanding than most people of the different little structures in the lungs and what happens with gas exchange and what happens with the little alveoli and where they are, and the whole 9 yards with that. And now you have a decent foundation in the basics of the immune system. You understand what a severe reaction from the immune system does, that it may be ramping up to kill a foreign invader, but then its own response is also damaging to the body. Okay? All right. That, I believe, was our last page. It was. Okay? So, there it is. We did it. Infectious disease. All right? If you have questions, comments, or of course to place your votes on the most disgusting word, mucus or sputum, please email me. And I will see you all soon. Why is my camera so fuzzy? There, not better. I'll see you soon.